The second microorganism group that we want to discuss is fungi. It is a multicellular organism except for yeast. Yeast is unicellular. It is eukaryotic cell. It composes cell wall that contain chitin. Fungi is such a diverse group. The study of fungi alone is helpful, especially when some of the fungi pose health risks to human as well as benefit to human where we use it for fermentation. The study of fungi is better termed as mycology. So fungi typically divided into two groups that we want to discuss. The first one is yeast and the second one is mold. Mold pose a health risk to human due to mycotoxin production. So it is a lot of concern, especially in dry food, because for mold it doesn't require a high moisture condition. The drier the condition will be more favorable for them to grow. These are some of the examples of useful yeast and molds that are commonly used to manufacture fermented food. The most common one that you may see here is bread. We use yeast to make bread, and basically that yeast is actually Saccharomyces cerevisiae. For cheeses, the blue vine here, if you can see it, is actually Penicillium roqueforti, or known as blue mold. It is used to manufacture blue cheese. So blue cheese usually have like a very distinct, very strong taste as compared to cheddar cheese or mozzarella, but some people like it, so that's fine. It's just that know that this is not type of spoilage. Is actually favorable, useful to manufacture blue cheese. Another one, maybe more common in Asian population, is actually soy sauce. Believe it or not, it's actually made from fermentation process where it uses a combination of mold and yeast as listed here. More traditional or more local food that use fermentation is actually tempeh, that use Rhizophos oligosporus, and also tapai, or known as fermented cassava. It uses a combination of diverse yeast and mold, or known as culture. The growth of fungi really related to an important component called thallus, which is the vegetative fungal body. So for yeast, it's actually considered as unicellular thallus because it replicates by itself. The layman term for asexual reproduction of yeast is yeast budding. So this figure here shows the yeast budding process. For mold, it's a little bit complex, which makes sense because mold is multicellular microorganism. It can grow via sexual or asexual reproduction. For thallus structure of mold, it contains a hyphae, which actually a tube-like filament structure that contains nucleus. So if there's a lot of hyphae, it is called hyphae, which is the plural term. And a network of hyphae, which is like a abundance of them, is actually formed like a mat of hyphae. So that called mycelium or mycelia in plural. So this figure is basically showing you the structure of hyphae and mycelium. So some fungus has spores, so this spore will germinate in form hyphae. And hyphae, like I mentioned before, is a tube-like filament structure, and when there's abundance of them, it will form a mycelium here. Another keyword for fungi that I want you to know is the septate versus aseptate hyphae. So basically, hyphae is this filament here. If it has like a section like this, it called septate hyphae. But if it's not, nuclear just laying continuously here, it is a septate hyphae. The pictures I'm showing you now is basically some of the example of mold contamination on food. The first three pictures here is showing you a different colors of mold. It can be blue, white, or green. So if you see this type of food, you obviously will avoid them at any cost because you can physically see them. However, most mold contamination is due to the presence of mycotoxin, which basically means that if these nuts or corn, for example, have been contaminated with a mold, and then you kind of peel the nuts shell, and then you just took the nuts, you may think that the nut is not contaminated, but we don't know how much mycotoxin production have been produced in such condition. So sometimes the manufacturer will kind of still retrieve this food and then process it to other products. Like for example, for corn, maybe they took some of the inferior quality of the corn and use it to make cereal, and then when you consume cereal that have quite a number of mycotoxin, it can produce health risks. Listed here is actually the mycotoxin and also food that is usually associated with this mycotoxin contamination. These are some similarities feature between yeast and mold that I want you to pay attention to. Both yeast and mold, or fungi in general, are obligate aerobes. They need oxygen. So all the food crops and also food that I show you in the earlier slide basically typically exposed to oxygen, which allow yeast and mold to grow. It can grow in broad pH condition, like the picture I showed you before. Strawberry have a different pH, maybe slightly acidic as compared to bread or peanuts. The growth temperature is usually just the ambient temperature, optimally 25 degrees Celsius. For mold and yeast, it requires lower water activity requirement than bacteria actually. But if you compare between the two, water activity of yeast usually a little higher than water activity of mold. Now we're going to get into the third group of microorganism, which is protozoa. So protozoa is microorganism, but it is also parasitic microorganism. Some of the example is listed here, and these protozoa usually associated with foodborne illness. Some characteristics of protozoa include unicellular microorganism. The size is larger than bacteria. It usually does not multiply in food because it needs a host for replication. So that's why it is parasite. Parasite usually need hosts to replicate. It doesn't replicate if it's in the environment or in the food. The infectious form of protozoa is cyst. Other parasites include helminths, or basically worms. There are two types of worm, which is cestodes, tapeworms, and also nematodes, roundworms. So it's actually a concern because the egg stage of this worm can be ingested by the cattle while feeding the grass, and then this egg can hatch inside the cattle and turn into larvae or oncospheres. Because it can reside in the muscle of the cattle, that means that if we consume undercooked meat, we actually can ingest this larva, and then this larva will grow in our intestine. This tapeworm is super long and it can live as long as we live. Because it's parasite, it will try to take advantage from the host by absorbing nutrient from the intestine. The way it attached to the intestine is using a scolex, which is a distinct feature of the tapeworms, as you can see in this figure here. Okay, now we want to talk about viruses. Virus is actually considered as a non-living particle because it's actually just a protein. It's actually a huge debate for within microbiologists, but the one that is acceptable at this current stage is non-living particle. The size is very small as compared to bacteria, and it can be RNA or DNA genetic material. Last but not least is prion. It is considered as infectious protein. So basically the history of prion is dated around 1980s, where in the UK it's a huge cases of mad cow disease. So when they figured it out, it's actually the cow has been fed with a cow carcass, which basically means that they're eating their dead friends. So based on that practice, the cow got a disease called BSE in scientific term, which is bovine spongiform encephalopathy, where they examine the brain of the infected cattle. This is how it looks like, a sponge-like appearance. So that's why the disease is called spongiform. That's where the name is originated. If we consume the infected cattle, we can get the neurological disease disorder. So what is this infectious protein? It's actually the misfolded protein that present in the central nervous system. However, the event that lead to the misfolded protein is actually not well understood until now. Similar disease can also occur in sheep, where it is called scrapie. In human, the unknown event that lead to misfolded protein occurring in human brain can cause disease called Creutzfeldt-Jakob disease or CJD. This disease can cause dementia for infected human. So from this we have covered the classification of microorganisms, and next we're gonna talk about factors affecting microbial growth.